In this video, we will discuss about SARS-CoV-2 virion cycle. This is the extension of virus cycle topic that we are discussing, and we have skipped one topic, that is virus replication and expression. So, as different families has different kind of process for virus replication and expression, we are taking the example of one of the recent virus that has caused pandemic, that is SARS-CoV-2. So far we have understand how the virus enters into the cell and how uncoating happens. So after uncoating, we will start from there. It is SARS-CoV-2 has genetic information in the form of positive single strand, let me write it, single strand RNA. So what is positive and its opposite one is negative single strand RNA. So the positive single strand RNA, the coding or you can say that or you can say that the nucleotides that are present in the positive single strand RNA are ready to be translated with the help of cell machinery, whereas in the case of negative single strand RNA, the virus that contains negative single strand RNA must have some enzyme that helps in converting those negative single strands RNA into the positive single strands RNA, so that the cell machinery can use positive single strands RNA for the used or not used. Cell machinery can translate those positive single strands RNA into the virus structure or the way the virus should replicate and express itself. So in case of SARS-CoV-2, it's a positive single strand RNA, so it is easier for the cell machinery to translate it. And it has, every RNA has 5' and 3' end. So in the 5' end, what it has two main open reading frame. Open reading frame. One is open reading frame 1A and open reading frame 1B. Okay. So what this open reading frame 1A, 1B does, this open reading frame 1A and 1B helps in the production of non-structural protein, NSP. It is called non-structural protein. What this non-structural protein does is, these non-structural proteins helps to form a virus replication, helps to form a virus replication complex. It forms around 15 non-structural protein, 15 to 16 NSPs, and out of this 15 are used to form a viral replication complex. 15 are used to form a viral replication complex. Okay. Apart from this, around most part of this open reading frame 1A and 1B comprises of you can say polyadenated and capped helps in the comprises of polyadenated and capped RNA. Helps or we can say it in different words like most part of this open reading frame has polyadenated and capped RNA. What is the function of this polyadenated and capped RNA? Is to stabilize the RNA and prevent it from degradation. So with the help of this, the RNA will survive for a longer duration of time. And what else we need to discuss? Yeah, and one more thing that we need to understand, it also this open reading frame okay so this is a brief introduction of what is there in the open reading frame 1A and 1B, and let us now understand what is there in the 3' end. 3' end has been able to transcribe proteins, transcribe structural 3' as structural protein codes, structural proteins. It has the RNA code for structural proteins near 3' end. At the same time, it also has RNA proofreading enzyme. As this coronavirus RNA is greater than 30 kilobyte, there is more possibility that during translation some mutation might occur that might kill the RNA. So in order to prevent those or reduce those chances, this RNA proofreading enzyme is present in the SARS-CoV2, so that it does not mutate in a way, or the chances of mutation are low, so in a way that it is harmful for the virus. So this is also present in the virus, but yeah. And so what happens? This thing 3' end also contains, or it's 3' end. Yeah. This the non-structural proteins. Now let's discuss something about non-structural proteins. A non-structural protein, as soon as they are translated, or we can say that during co-translation also, and during translation, after translation, it starts to form a membrane, double membrane, convoluted membranes kind of thing that will protect the virus, that will protect the virus. It's like a envelope or nucleocapsid things. It will form a membranes like double membrane, convoluted membrane, and there are other things also that will prevent the virus when it moves into the extracellular matrix, so that it can infect the other cells. Prevent virus from prevent virus in extracellular matrix, prevent virus from degradation. You can say that, degradation. So when the structural proteins are formed, it will then go to endoplasmic reticulum, and from endoplasmic reticulum there is post-translation modification occurs, then it moves to the Golgi, as we have discussed this, and glycosylation pathway. And this, as we know that this virus particles, most of the virus or some of the virus has nucleocapsid also. So nucleocapsid RNA, when it interact in the Golgi, the budding process occur. Interact in the Golgi, the budding process occur by that the budding process a vesicular with the because of this budding process. Now the RNA or the we can say that the SARS-CoV2 virus is now ready for translated into the is ready for translated into the extracellular matrix to infect the other cells, So in this way, these are some of the important points of the SARS-CoV2 virus. Okay, so next video we will discuss what are the strategies that has been used to make this SARS-CoV2 virus ineffective or less reproducible. Thank you.